Hi everybody, welcome back. We're going to be talking about eukaryotes today. And as you can see from the cartoon, eukaryotes can exist either as unicellular organisms, meaning they have one cell, or multicellular organisms. We've already talked about two of the domains. We've talked about bacteria and archaea. Both of those are prokaryotes, which means they don't have a nucleus or membrane-bound organelles. Today we're talking about the eukarya domain. The eukarya domain has 4 different kingdoms within it. As microbiologists, we don't really talk about plants, so in this section of the course, we're only going to be talking about the protista, the fungi, and the animalia. This is a picture of a generalized eukaryotic cell. If you look at the list on the right, you can see a partial list of structures that you would find in all eukaryotes. You are responsible for knowing the structure and function of each of these items. In the list on the left, you can find things that are found in some eukaryotes. Some of them have them, some of them don't. So for example, a flagella is usually used for locomotion. In human cells, we do not have flagella except one specialized cell, which would be the sperm cell. We're going to be going through each of these in more detail. We'll start with the outside of the cell with cilia. Cilia are not found in all eukaryotes. They're only found in a single group of the protozoa, which are part of the protista kingdom, and certain types of animal cells. They're not found in prokaryotes, fungi, or plants. They're similar in structure to flagella in that they wave. However, when you see cilia, they're usually much shorter and more numerous. You can have up to 1,000 cilia on one cell, whereas with flagella, you only have a few at most. And they have different functions depending on what type of cell they're on. So in a multicellular organism, like in the animal kingdom, cilia move fluid or objects across the surface of the cell. One place you would find cilia would be in the uterine tube, and they would beat to help push the egg down from the ovary into the uterus. In unicellular organisms, cilia are there to help move the cell around. So the cilia all stick out from the cell, and they're like little oars. They row the cell about. Also outside the cell, in some eukaryotes, is the glycocalyx. This is also sometimes called an extracellular matrix, and this is made up of long strings of sugar molecules. It has a couple of different functions. The glycocalyx helps to protect the cell from predation. It helps it to stick to one another, and it also helps in the reception of chemical signals. Some eukaryotes also have a cell wall. These are found in fungi and algae, which is part of the protista kingdom. The cell wall is rigid and it helps to provide support for the cell. The cell wall in eukaryotes is very different than the cell wall in bacteria. It's not peptidoglycan. Remember, peptidoglycan is what bacterial cell walls are made of, and it's specific to bacteria. So the cell walls in eukaryotes are made of different things. The plasma membrane is found in all eukaryotes, and it's made out of phospholipids. Remember that a phospholipid is made out of a hydrophilic phosphate group at one end, and the other end are 2 hydrophobic fatty acid tails. This allows the plasma membrane to spontaneously form into the fluid mosaic model, where the charged phospholipid heads are on the outside and the hydrophobic core is inside. The nucleus is where our genetic material is stored. It is separated from the cytoplasm by the nuclear envelope. This is a double-layered membrane, so you have 2 bilayers. And again, remember how important our DNA is to the survival of our cells. So we want to protect them with 2 layers of plasma membrane. There's an area inside the nucleus called the nucleolus. It's very dark staining. This is where ribosomal RNA synthesis happens. So this is where ribosomes are made inside the nucleus. We also have the chromatin, and the chromatin is DNA and proteins all wound up and wrapped up together. Remember when we talked about prokaryotes, we talked about the DNA being circular. In eukaryotic cells, the DNA is linear. You can have lots of pieces of DNA that are wrapped around histones and other proteins, and this makes up our chromosomes. Ribosomes are another structure that are found in both eukaryotes and prokaryotes. However, in eukaryotes, the ribosome is bigger. The eukaryotic ribosome is 80S, whereas the prokaryotic ribosome 70S. You can find ribosomes throughout the cell. They're made in the nucleolus of the nucleus. They leave the nucleus and go into the cytoplasm. They can stay in the cytoplasm or they can become attached to the rough ER. You can also find ribosomes inside the mitochondria and chloroplasts. The function of ribosomes is to make protein. The endoplasmic reticulum is a series of membranes that are continuous with the nuclear pores. So messenger RNA that's made in the nucleus can leave through the nuclear pore and directly enter the endoplasmic reticulum. And there are two types of endoplasmic reticulum. The first one is called rough endoplasmic reticulum, or rough ER. And the reason it's rough is because it has ribosomes attached to it. So remember the function of ribosomes? It was to make protein. So the function of the rough ER is going to be protein synthesis. After the proteins are made, are synthesized, the rough ER is going to deliver those proteins to the Golgi in vesicles that bud off from the rough ER. The other kind of ER is called smooth ER, and it's smooth because there's no ribosome. The function of this type of ER is synthesis and storage of lipids, as well as detoxifying the blood. You can see different amounts of each type of ER in different types of cells. So in your liver cells, for example, you're going to have lots of smooth ER because it's going to be detoxifying the blood. In your muscle cells, however, you'd probably have more rough ER because those cells are having to make a lot of protein. The Golgi apparatus is the post office of the cell. Molecules that come to the Golgi are modified, sorted, and sent out from the Golgi to different parts of the cell. There are two faces of the Golgi. The cis face is facing the endoplasmic reticulum. So the vesicles that are leaving from the ER go towards the cis face of the Golgi and fuse with it. Anything inside that vesicle is released inside the Golgi. The trans face of the Golgi is on the other side, and this is where vesicles bud off. And they're generally going to one of two places. They're either going to be taking digestive enzymes to the lysosomes, or they are secretory vesicles, in which case the things that are inside the vesicles are going to be released to the outside of the cell after fusing with the plasma membrane. So the secretory pathway of the cell involves the nucleus, the ER, and the Golgi, and it uses vesicles to move product from one place to another. So if you start in the very beginning, inside the nucleus, the ribosome is made in the nucleolus, and messenger RNA that has been transcribed from DNA also starts in the nucleus. Both of them are going to leave the nucleus through the nuclear pores. The messenger RNA gets translated by the ribosomes into protein, and this happens on the rough ER. After the protein is made, it will be sent to the Golgi in a vesicle that buds off from the rough ER and then fuses with the Golgi on the cis face of the Golgi. The proteins that are inside those vesicles will then be modified and sorted and packed into more vesicles. Since we're talking about secretion here, all of these proteins are destined to be released outside of the cell. So the vesicles will move from the trans face of the Golgi to the plasma membrane and out. Phagocytosis is a process that's the opposite. This is where we're taking things in from the outside. And since it's phagocytosis, we're talking about a foreign particle being brought in. The first thing that happens is that the foreign particle is engulfed by a piece of invaginated plasma membrane. This is called a phagosome. You can see the particle inside a little vacuole. The lysosome is going to merge with the phagosome. And remember, the lysosome is filled with digestive enzymes. It's going to break down anything inside that phagosome. Once the foreign particle has been broken down, the cell can decide what to do with the pieces. The mitochondria is known as the powerhouse of the cell, and this is where organic compounds are converted into usable energy, or ATP. Mitochondria have their own DNA, but the DNA in the mitochondria is circular. The mitochondria can divide independently of the cell, so you can make much more mitochondria if you need them without actually having to make more cells. The cytoskeleton of the cell has several different functions. The first one is it's going to help anchor everything in its place so things aren't just randomly bumping around. Parts of the skeleton are also important in moving vesicles and RNA from place to place. And in addition, the cytoskeleton helps to either hold a certain shape or allows the cell to change shape and move around. There are 3 types of filaments associated with the cytoskeleton: microfilaments, which are the smallest, intermediate filaments, which are the middle, and microtubules, which are the biggest ones. So as a review, we have 3 domains that we deal with in microbiology, and of those, 2 of the domains are prokaryotes and 1 is a eukaryote. In the eukaryotic domain, we have 4 different kingdoms that you're going to be working on in class. That's it for today. See you in class.